Hello, everyone, and welcome to today's webinar, Catapult Your Cell Therapy Research into the Clinic Beyond Immuno-Oncology. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit www.miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. Our speakers will answer questions via email to the address you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Dr. Chao Sheng, expert for cell culture and regenerative medicine, Miltenyi Biotec. Dr. Viola Stella Palladino, expert for regenerative medicine, Miltenyi Biotec. And Kirsten Langeveld, Global Product Manager, Miltenyi Biotec. Speakers, you may now begin your presentations.
This webinar about pluripotent stem cells culture and expansion from research to clinical applications. So today, me, Viola Stella Palladino, and my colleague, Chao Sheng, will introduce you to Miltenyi Biotec latest solutions for culture of pluripotent stem cells. So we will see first solution for classical manual expansion and handling of these cells. And then we will introduce you to our instruments for automated and GMP compliant PSC manufacturing. So in the last decade, the implementation of new type of cellular models has opened a whole new world of possibilities, both for research, but also for clinical applications. In fact, induced pluripotent stem cells, also known as iPS cells or iPSCs, can be generated directly from an already differentiated somatic cells. Then they can be reprogrammed into a pluripotent state and can be further differentiated into any cell type of the human body. This, of course, overcomes all the limitation of working with cell types that would be otherwise extremely difficult to obtain by classical means. In the last decades, the pluripotent stem cell field has seen enormous advances, and these cells find now important applications in the study of the etiology of diseases, in cell replacement therapies, as well as cellular models for drug discovery and toxicity screening. But independently from what is the final application of these cells, when working with pluripotent stem cells, it's extremely important to have an established workflow for their culture in an undifferentiated state. As we will see in a few moments, a key factor to ensure the high quality of the line is the media that we use to culture our cells. So for this reason, today we will focus on culturing media that can be used for culture of PSCs under xeno and feeder-free conditions. Why is it so important? Well, this is because pluripotent stem cells are very sensitive cells, and incorrect handling or using unfitting reagents can lead to the loss of their pluripotent characteristic. And this is a major concern because just high-quality PSC lines are then suitable for experimental applications. So it's clear that having a robust and stable culture environment is a fundamental requisite for successful culture and therefore successful experiments. Having this concept well in mind, we have developed a new generation of cell culture media for PSC maintenance and expansion, StemMACS PSC Brew XF. StemMACS PSC Brew supports, in fact, consistent cell growth without compromising the maintenance of stem cell-specific characteristics. But which are these characteristics that we should monitor for? First of all, a good media allows PSCs to maintain their typical morphology. And here you can see that in these pictures of pluripotent stem cells that have been cultured with PSC brew. So as you may see, cells cultured with our media maintain all the key morphological characteristic of pluripotent stem cells. Moreover, they also display classical and consistently stable doubling times over consecutive passages. A second important criteria that we should always keep in mind is that a robust media must support and allow high and actually persistent expression of pluripotency markers, also when PSC are maintained in culture for a longer time. And here you can see the assessment of this feature by flow cytometric analysis of pluripotency associated markers at different passages. As you can see from the graphs, the lines display high and persistent expression of TRA160, CA4, OCT34, SOX2, and these are all markers that are associated with a pluripotent state. And on the other hand, they showed always a very low expression of CA1, that is an early differentiation marker. At the same time, the media should also allow the cells to maintain their full pluripotency, also on a functional level. And the media should not introduce any bias in the differentiation capabilities of our lines. So we assess this feature by testing if PSC lines cultured in CliniMACS PSC brew can differentiate into cell types that derive from the 3 embryonic germ layers. As you might already know, the 3 embryonic germ layers, so endoderm, ectoderm, and mesoderm, form really early during the development, and then with time, they will give rise to all the cells that compose the human body. So to assess if our pluripotent stem cells maintained in this condition would maintain also these pluripotent characteristics, we used a specific kit, the CliniMACS 3 lineage differentiation kit. And this kit allows to induce the cells to differentiate into the 3 germ layers if they are actually pluripotent. Then we analyzed the results by flow cytometry that confirmed the ability of these cells to differentiate into mesodermal lineage, into definitive endoderm cells, and into neuroectodermal cells. Finally, we know that PSCs tend to acquire genetic aberration if maintained in prolonged culture. And this is true especially if they are mishandled. The genomic stability of the cells maintained in our media was analyzed by karyotyping and also more in detail by evaluating the burden of copy number variants present. And in both cases, we see that the karyotype was stable also after long-term cultivation, so cultivation for more than 10 consecutive passages. Okay, so after talking about important features of a culture media for the cells, let's add also something that is important for the researchers. So as just mentioned, PSC culture have distinctive metabolic demands and do not tolerate stress conditions. And this is the reason why the media changes have been traditionally performed with 24-hour interval. But by using new generation cell culture media, such as CliniMACS PSC brew, more flexible feeding schedules can be applied. So you can either choose the classical everyday feeding, so every day changing the media, or you can opt for every other day feeding, or even nicer, you can skip feeding on the weekends. How did we achieve this flexibility in feeding schedules? Well, it all comes down to the ingredients that were used for this media. So, for example, in the new formulation, we have a stable FGF2. This is an improved sequence FGF2 that has a higher thermostability as well as a higher resistance to proteases, while it maintains the same biological properties as a natural FGF2. And this, of course, ensures that FGF2 is present in the culture in all the feeding schedules that we want to apply. Additionally, we also have carefully improved the buffering capacity of this media. And as you can see, the CliniMACS PSC brew ensures pH values within acceptable range in every feeding schedule applied. We have also evaluated the level of nutrients in all the strategies. So as you know, carbohydrates in the form of sugar, so for example, glucose, are one of the major sources of the for energy for the cells in culture. And as you can see, the levels of glucose decrease physiologically because the cells are of course using it, but we do not see differences due to the different feeding schedules that we apply. Similarly, the levels of glutamine, that it's of course an essential amino acid that it's also required for the proliferation of the cells. So the level don't really show any significant difference due to the feeding schedule. Finally, we have evaluated the presence of byproducts in the culture. So as you might know, lactate accumulation in the media occurs naturally, but when we have very high levels, this might reduce the cell growth and productivity. And similarly, in aqueous solutions, such as the culture media, the glutamine will spontaneously decompose into ammonia. And also ammonia has been reported to be toxic and inhibitory for mammalian cells. So we have evaluated the concentration of these two byproducts in all different strategies, and we report that there is no significant difference due to the strategy itself. So this concept further strengthened the fact that the media can be used in a very flexible way to fit the need also of the researcher. One last feature of this new culture media is that, of course, we know that the field is evolving and that in the future, more and more application will take advantage of pluripotent stem cell line. And we want to enable the researchers to be able also to perform this more challenging application. So, for example, gene editing or cell sorting. Therefore, we have developed a special supplement, the CliniMACS PSC support, that is fully compatible with our culture media. And here you may see how these two media have been combined to support the growth of TRA160 CA4 positive cells that have been sorted using our MAX-Quant title and then plated in low density in a 96 well plate. So after 10 days of culture, 6 clones were chosen and further expanded. We then perform, of course, quality control of these cells to see if they maintained all the pluripotency characteristics that we have seen before, and all the cell lines shown to be highly pluripotent and could be used for further differentiation into different cell types. So in this first part of the presentation, we saw how choosing a good culture media for pluripotent stem cells is a key point in successfully culturing and maintaining these cells. Let's now see what solution we could apply if we want to move one step further and manufacture PSCs in an automated and closed way. And for this, I leave the stage to my colleague, Chao.
Okay, thanks a lot, Viola. Really welcome to our webinar from my side. And I'm really happy to really share our latest experience and our workflows with you more this time on the manufacturing of PSCs, more for the clinical applications. So as I think we all know that really right now it's a very exciting time for pluripotent stem cells, as we see many scientists are already using trying to translate their research experience and their expertise into clinical applications to really address many devastating diseases. So today I will very quickly show you some our established workflow at Miltenyi Biotec for automated, closed, and GMP-compliant manufacturing of human pluripotent stem cells that might be, very helpful and supportive for your clinical translations. Well, speaking of manufacturing of pluripotent stem cells, PSCs, and their derivatives for these clinical applications, so we all know that these are very challenging and very complex endeavors, and most of these current manufacturing processes involve multiple expensive instruments, very skilled operators, and also high-class clean rooms. Meanwhile, standardized in-process control and quality control assays are also required throughout the entire process. So, and also in some cases, like the users also wants to somehow isolate the desired cell populations for downstream applications that will also require a sterile, gentle, and GMP compliant isolation technique. So all these factors together could really limit the manufacturing skill, compromise the reproducibility, while at the same time also significantly drive up the cost. So, I mean, everyone in this field is facing the same question, the same challenge. That is, how to make these complex processes more standardized, more scalable, also at the same time more affordable for the end users. And here are some solutions from Miltenyi Biotec. So with the CliniMACS Prodigy cell manufacturing platform and all these separated open handling steps, multiple devices, the skilled operators, the high-class clean rooms, they can all be integrated into one automated and closed system. And the users can also build customized cell manufacturing workflows, including for the PSC expansion differentiation using the CliniMACS Prodigy. And then with the MaxQuant analyzer, so then standardized flow cytometry IPC-QC assays can be realized with the automated technologies. And finally, with the MaxQuant title cell sorter, so this can be applied to isolate the desired cell populations with a very gentle, fast, and also GMP compliant cell sorting technology. So here today, due to the time limit, so we will focus on the CliniMACS Prodigy. So together with the single-use tubing sets, actually the CliniMACS Prodigy allows automated cell processing from starting materials to final products. And upon your request, so the Prodigy can actually automate all these different cell handling steps, including sample preparation, dense degraded centrifugation, cell washing, medium change, etc., up until the final formulation step. And actually, the CliniMACS Prodigy platform is very open and very flexible, and we have already developed a very broad range of software applications for various cell types and various workflows. For example, for the CAR T cell manufacturing processes. Well, for the adherent cell types, like pluripotent stem cells and their derived somatic cell types, now we have this adherent cell culture process to address these requirements. So the CliniMACS Prodigy adherent cell culture process actually allows cell cultivation to take place in two different units, the centrical unit and the external culture vessels. So first we have the centrical unit, and there's a CliniMACS Prodigy chamber lies inside this unit, where you can support you can do adherent and non-adherent cell cultures. You could also automate the cell feeding, cell harvesting within the centrical unit, and you can control, of course, the temperature, the concentrations of CO2 and air. There are also a camera integrated under the chamber so that you could visualize your cell manufacturing, your cell cultivation, and there are also multiple input port available so that you can supply the cell cultures with different medium and cytokines. And this chamber actually offers a 100 square centimeter surface space. But we know that for PSC expansion differentiation, very common that you would need a larger surface space. So for this, you know, external culture vessels can also be easily connected to the CliniMACS Prodigy tubing sets via stereo welding, so to enlarge the cell culture space. So for example, these multi-layer cell stacks can be very easily connected to the tubing set. And right now we have tested that you can actually connect maximally 3 of these 5-layer cell stacks at the same time to the tubing set. And with that, you will have roughly 10,000 square centimeter surface space. So it will be fairly large for the cell culture. And then with the when we designed this adherent cell culture software, actually our goal is really to make sure that it's very flexible, it's flexible enough to culture different cell types. That's why we actually modularize the individual cell handling steps. For example, now on the main menu, you can find these different modules for density gradient centrifugation, for coating, for inoculation, medium change, harvesting, etc. And the users can very freely select and combine these different modules. And as a result, so very complex workflows like direct differentiation of PSCs can be realized, can be adapted into the closed and automated CliniMACS Prodigy adherent cell culture process. And within each module, the users can also set up all the parameters according to their established SOPs. For example, in the coding module, the users can set up the coding temperature, the coding time, the volume of the coding reagents, etc., all according to their established procedures. So we have already established some workflows for GMP-compliant expansion of human MSCs and PSCs. We also have established workflows for directed differentiation of PSCs into brain dopaminergic progenitors and also cardiomyocytes on the Prodigy. So today we will focus on the GMP-compliant expansion of PSCs on the Prodigy. So for this, we have first developed the iPSC Brew GMP medium, which is manufactured and tested under the Quality Management System ISO 1345, and is actually also designed following the recommendations of USP 1043 on ancillary materials. So it is currently available in bottles, but very soon, then we will also offer this GMP compliant medium in bag format, and that will be a great match with the CliniMACS Prodigy for closed PSC expansion. And in our experiment, as you can see, we showed in this result, so this GMP medium enables very stable and very efficient human PSC expansion as both clusters and also as single cells. And this medium also supports very fast culture reinitiation after the cryopreservation. So this is a overview of the GMP compliant PSC expansion on the CliniMACS Prodigy using the iPSC Brew GMP medium. So we have a dedicated application sheet. So here I will not go into details, but as you can see already on this, so here you can apply different modules for coating, for inoculation of PSCs, for culture, for medium change, and as a result, so all these steps can be automated within the CliniMACS Prodigy adherent cell culture process. And we have also performed side-by-side comparisons between manual and Prodigy-based PSC expansion using our Reinfinity antibodies and also using the StemX trilineage differentiation kit. And looking at the expression levels of these potency markers and also the trilineage differentiation potentials. And we can see that the Prodigy versus manual control, they really gave rise to very comparable results. So by using the centric unit and the different sizes of the external culture vessels, the users can actually very easily reach different PSC expansion skills using the CliniMACS Prodigy and with much less open handling steps in comparison to the manual process. We know that establishing GMP-compliant PAC banks is a very important step in many clinical applications. And for this purpose, the closed and automated CliniMACS Prodigy adherent cell culture process, together with the iPSC brew GMP medium and our standard QC reagent, so they could be very valuable assets for the GMP-compliant PAC expansion and banking purposes. So here, just a very quick example. Very recently, Dr. Raphael Crow from Syra Foundation in Japan has successfully actually transferred their manual clinical-grade iPSC expansion workflow into the CliniMACS Prodigy adherent cell culture process. And he has presented his great work at the IACR Tokyo Symposium and also at a joint webinar with us last year. So actually, that's already everything from my side as well. Thanks a lot.
Hello, welcome everybody to this webinar on T cells and COVID-19, translating research findings into clinical options. My name is Kirsten Langeveld. I am a product manager for ex vivo T cell depletion systems for the productions of cell therapy products. But before we start, please acknowledge the displayed disclaimer. In the first part of today, I will briefly introduce products and workflows that have been optimized to study T cells in SARS-CoV-2 infections. In the second part, I will introduce the product systems of Miltenyi Biotec for the generation of clinical-grade cell products and their use in published cell therapy concepts to fight SARS-CoV-2 infections. Why is it important to study the role of T cells in COVID-19 infections? T cells play a key role in the defense against viral infections, but also in the generation of immunological memory, which will give protection from future infections. Studying T cells is therefore important for research on evaluating immune responses and to investigate options of an adoptive cell transfer for future therapeutic purposes. Let us have a look on tools and technologies to study antigen-specific T cells. So first, T cells need to be isolated from a donor sample. Here, for tissue dissociation, the GentleMACS tissue dissociator is an appropriate choice. Next, the cells are stimulated with the antigen of interest. Please note that antigen-presenting cells need to be present to efficiently activate the isolated T cells with a certain antigen. When working with PBMCs, this is already the case. However, with isolated T cells, APCs have to be added separately. Following stimulation, only the antigen-specific T cells will get activated, and the cells can then be stained intra- and extracellularly to assess their phenotype and analyze cytokine expression. Before cell analysis, antigen-reactive cells can also be magnetically pre-enriched using distinct surface markers. And then, finally, the cells can be analyzed using flow cytometry, for example with our MaxQuant flow cytometer, or sorted on the MaxQuant Taito cell sorter. Here, the efficient antigen-specific stimulation of T cells is achieved with our Peptivator peptide pools. Peptivator peptide pools efficiently stimulate both CD4 as well as CD8 positive T cells, and we cover an extensive portfolio of virus, tumor, fungi, microbiota, and autoantigens. The peptide pools consist of 15 mer peptides with 11 amino acid overlaps, which cover the complete sequence of the respective antigen. Our Peptivators are available in research, premium, and even Max GMP grade, which allows a rapid translation in the clinic. Finally, they are also available in the convenient 96-well format for high throughput applications. So our Peptivators can also help to standardize your T cell assays, as they are ideal simulation reagents for antigens and very specific T cells. Furthermore, I want to point out the cytokine secretion assays of Miltenyi Biotec. See, cytokine secretion assays allow the viable enrichment of cytokine secreting cells from mouse and human samples. The principle is rather simple. First, the secreted cytokines are immobilized on the cellular surface by the so-called CAT reagent. Basically, thereby, the cell labels itself with its own cytokines. In a second step, the immobilized cytokines are then labeled with fluorophore-conjugated antibodies, called detection reagents. Living cells can then be detected without intracellular staining using flow cytometry. In an optional third step, the fluorophore-labeled cytokine-secreting cells can be specifically enriched and used in downstream assays with the help of anti-fluorophore magnetic beads. Consequently, our cytokine secretion assays come in two versions. For flow analysis only, Miltenyi offers the various detection kits, while for enrichment and analysis of viable cytokine secreting cells, we have several enrichment and detection kits. For the isolation of antigen and virus-specific T cells in a clinical setting, we offer a clinical-grade cytokine capture system running on our CliniMACS Prodigy platform. For clinical-grade production of cell products against COVID-19 infections, the Peptivator SARS-CoV-2 Select is already available in Max GMP grade. In addition, the CliniMACS system can also be used for the depletion of CD45 array positive T cells to produce cell products that contains memory T cells. So let us now switch to the news from the field on clinical applications. Cell therapy products based on virus-specific donor T cells have been used to treat opportunistic viral infections in the context of allogeneic hematopoietic stem cell transplantation. It has been discussed whether these treatment options could also be applied to COVID-19 patients. In this context, I would like to present the work of three different groups from Singapore, the United Kingdom, and Spain. As the authors explain in these publications, the transient presence of donor T cells in COVID patients could be possible under certain conditions. On the other hand, it would be imperative to prevent alloreactive reactions against the patient. Miltenyi Biotec offers workflows for the production of cell products that are used in certain cases to prevent or treat opportunistic infections in patients with allogeneic HSCT. In the publications, the authors describe how these workflows could be used to produce cell therapy products against SARS-CoV-2 infections. First, some background information on T-cell-based therapy against infections. This treatment was originally developed in the context of allogeneic HSCT. Until the reconstitution of their new immune system, these patients are at high risk for opportunistic viral infections as CMV, ADV, EBV, or BKV. Antiviral drug-resistant cases have been treated with adoptive T-cell therapy. Miltenyi Biotec provides tools for manufacturing T-cell-based products that are used to treat certain viral infections in allogeneic HSCT. For 17 years, the clinical application of these treatment options has been continuously published in peer-reviewed journals. By now, new ways to treat COVID-19 patients with adoptive T-cell therapy under certain conditions have been described. Let us start with the concept of transferring virus-specific T cells to combat certain viral diseases. As reported, adoptive transfer of allogeneic VSTs can rapidly restore virus-specific immunity and control viral infections after allogeneic stem cell transplantation. Sources of virus-specific T cells may include stem cell donor-derived VSTs in the setting of allogeneic stem cell transplantation, and third-party donor-derived VSTs, which has also been used in patients with infections after solid organ transplantation. The production of antiviral T cell products requires the enrichment of relevant VSTs and depletion of potentially alloreactive T cells. There are several strategies to produce these products, as for example those that are based on ex vivo expansion. Another option is provided by the CliniMACS Prodigy CCS interferon gamma system from Miltenyi Biotec. For this option, it is necessary to find a convalescent donor for the desired viral target. How does the CliniMACS cytokine capture system work within the CliniMACS Prodigy process? The CliniMACS Prodigy process integrates all critical steps of the CCS workflow, from antigen-specific T-cell stimulation to labeling of target cells, magnetic isolation, and formulation within 12 hours of total time. Importantly, all cell processing steps are automated, ensuring a convenient and highly standardized separation process. These include stimulation of the starting product from a convalescent donor with a virus-specific peptide to activate the desired virus-specific T cells, separation of these cells via the interferon gamma secretion, and the magnetic separation strategy to enrich interferon gamma-producing cells. Recently, we launched a MAX GMP peptidator targeting SARS-CoV-2 specific peptides. And we will be glad to provide the reference list with clinical applications. All necessary consumables for these procedures are supplied by Miltenyi Biotec. The CliniMACS Prodigy CCS interferon gamma system is available as a CE-marked medical device application. Outside the EU, regional legislation and regulations are applied. The clinical use of the cell product is subject to regional legislation and is under the responsibility of the user. Let us now take a look at the second possibility of producing cells against certain infections. Donor-derived CD45-A-depleted cell products have been used in the field of allogeneic HSCT. Memory T cells provide immunity against many viral infections that the donor has experienced during his lifetime. Prior to clinical application, it was shown in mouse models that the alloreactive potential is high in the naive T cell compartment and significantly reduced in the memory T cell compartment. CliniMACS CD45 depleted cell products are used to treat opportunistic infections and to enhance immune reconstitution in the setting of allogeneic HSCT. CD45 is an isoform of the CD45 family that is present on 100% of naive T cells, but not on CD45RO positive central and effector memory T cells. It is also present on approximately 100% of B cells, 80% of NK cells, and 30% of monocytes. CD45 is not a typical T cell marker, but it is an ideal marker to deplete alloreactivity from a product in one step, leaving specific memory T cells with the given benefits, and thus represents an excellent production option for clinical applications. Miltenyi Biotec offers two technical options for the clinical grade depletion of CD45 positive cells from leukapheresis products, performed on either the CliniMACS Plus device or the CliniMACS Prodigy device. The starting fraction is labeled with antibodies targeting the surface marker CD45 Magnetic separation results in a target cell fraction passively enriched for memory T cells. The CliniMACS Plus CD45 system has already been released to market in 2011. Please contact us if you would like to receive the full list of references for this clinical application. Consumables for these procedures are supplied by Miltenyi Biotec. Two CE-certified medical device applications are available for the depletion of CD45 array positive cells. The CliniMACS Plus CD45 array system provides a semi-automated procedure for starting products, containing up to 50 times 10 to the 9 white blood cells. The CliniMACS Prodigy PP45 array system provides a fully automated process for smaller cell amounts in the starting material. In addition to leukapheresis products, whole blood can also be used as starting material with this system. Outside the EU, regional legislations and regulations are applied. The clinical use of the cell product is subject to regional legislation and is also under the responsibility of the user. Several groups have developed manufacturing processes for cell products from convalescent donors targeting SARS-CoV-2 infections. We present here 3 publications in which these new developments are described. They are all open access papers. Bing Liang and his co-workers describe a process for flexible and rapid manufacturing of virus-specific T cells once a donor is available. This option uses the CliniMACS cytokine capture system, interferon gamma. The publication from Cooper et al. Describes a process that also starts with the enrichment of virus-specific T cells using the CliniMACS cytokine capture system. The difference to the previous publication is that here a culturing step is added to increase the harvest of virus-specific T cells. The final products can be provided within a biobank. The third publication by researchers from Spain also describes the generation of cell products for biobanking. However, here, memory T cell products are provided that are generated using the CliniMACS Plus CD45 array system. Infusion of these cells in COVID-19 patients has been previously published by Antonio Perez and his collaborators. I would now like to briefly present these important publications. However, due to the large amount of information contained in these publications, I will only briefly touch on selected points. To get a complete picture of these important projects, I recommend reading the original publications, which are all open access publications. The two objectives of the study by Wing Liang and collaborators were to investigate whether sufficient numbers of SARS-CoV-2 specific T cells are present in the blood of convalescent donors. Another important point was to investigate whether it is possible to produce clinical-grade products overnight. Whole blood or leukapheresis products were collected from the convalescent donors, and buffy coats were prepared from the whole blood samples. Leukapheresis products and buffy coats were stimulated with overlapping peptides of SARS-CoV-2 specific S, N, and M proteins. The stimulated products were processed by CliniMACS cytokine capture interferon gamma procedures, and the resulting fractions were analyzed for the amount of interferon gamma producing cells, their phenotype, T cell receptor V beta specificity, and functionality. The enrichment procedures resulted in final products that contain a median of almost 1 × 10⁶ interferon gamma positive T cells. To demonstrate functionality, one of the samples was successfully re-stimulated with peptide-loaded dendritic cells. Subgroup analysis revealed a relative depletion of naive T cells and an enrichment of effector memory T cells in the enriched fraction, suggesting that alloreactivity was reduced. T cell receptor V beta spectral typing showed markedly high oligoclonal peaks in the final products. They also concluded that overnight production of clinical-grade SARS-CoV-2 specific T cells is feasible for therapeutic purposes. The alloreactive potential was reduced while virus-specific T cells were retained in the product. They also suggested a dosage of 5 × 10⁶ SARS-CoV-2 T cells per kilogram based on previously administered cell numbers in other clinical settings. Under this assumption, 1 unit of whole blood could be sufficient to produce 3 to 6 products for adult recipients. The authors note that, based on previous studies by other authors, virus-specific T cells from donors who shared only one HLA molecule are effective. Based on this information, for the 6 donors in the study, the probability of potential patients having at least one HLA match would be more than 88% for Caucasians, more than 95% for Chinese, more than 97% for Malays, and more than 99% for Indians. All these and much more details can be found in the original publication as given below. The group of John Campbell and his collaborators is featured in the forthcoming publication by Rachel Cooper et al. The goal of this study was to characterize SARS-CoV-2 peptide-specific memory T cell populations in convalescent donors. In addition, the authors aim to investigate the feasibility of isolating and expanding these T cells on a clinical scale using an integrated cell culture step. The experimental design included scale-up experiments, functional testing of the obtained cells, and experiments comparing blood from convalescent donors with blood from donors who have not undergone SARS-CoV-2 infection. Important findings of this study are that generation of interferon gamma responses in SARS-CoV-2 virus-specific T cells from convalescent donors is possible after peptide stimulation, isolation, and expansion. Another important finding was that the presence of HLA class 1 and 2 alleles is widespread in the UK population, and that some specific HLA subtypes with strong peptide presentation ability were identified. Finally, the authors concluded that their method for generating large amount of virus-specific T cell products is feasible for clinical trials, supporting severe SARS-CoV-2 infections with comprised endogenous T cell responses. Here we can get an idea of the production method developed by John Campbell and his collaborators in Edinburgh. To begin with, a leukapheresis product is obtained from a convalescent donor. The V's teeth contained there are stimulated and enriched with the CliniMACS Prodigy CCS interferon gamma system and appropriate SARS-CoV-2 specific peptides. The harvested SARS-CoV-2 specific T cells are then cultured for 21 days with irradiated cells from the non-target cell fraction as feeder cells. As indicated, the final cell product contains between 10 to the 10 and 10 to the 11 cells. Restimulation experiments with dendritic cells demonstrated virus-specific T cell functionality after cultivation. The final product is then frozen and can be stored in a biobank system. After 14 days of expansion, central memory CD4 positive cells are the predominant subtype. Furthermore, and as stated, the expanded virus-specific T cells showed negligible co-expression of T cell exhaustion markers PD1 and TIM3 in both the CD4 and CD8 compartment, indicating the cell culture expansion has not induced an exhausted T cell phenotype. All this and much more details can be found in the original publication, and you find the information below. Antonio Paris and his collaborators from Spain have developed a different method for delivering cell therapy products to COVID-19 patients. They use CliniMACS CD45 array depletion to generate products that are stored within a COVID-19 biobank from convalescent donors. An important question was whether SARS-CoV-2 specific memory T cells exist in convalescent donors and whether they can be enriched by CD45 array depletion. This was shown in experiments with PBMCs from uninfected and convalescent donors. In stimulation experiments with three peptides, S, N, and M, interferon gamma secreting CD45 RO positive memory T cells were detected in convalescent donors but not in uninfected donors. A clinical scale product was generated using the CliniMACS Plus CD45 array system. Again, interferon gamma secreting CD45 RO positive memory T cells could be generated in the target cell fraction. To check functionality after freezing and thawing, the final product was frozen, then thawed and stimulated with EL15 for 72 hours. And the subsequent phenotype assay revealed a phenotype characteristic of an activated stage. Here we can get an overview of the production process. A leukapheresis product was obtained from a convalescent donor. As reported, after a CliniMACS Plus CD45 array depletion procedure, 80% of the cells were CD45 array negative T cells. About 84% of the T cells were CD4 positive, and about 14% were CD8 positive. After simulation with SARS-CoV-2 specific peptides, the percentage of interferon gamma secreting cells in the T cell subsets ranged from 0.31 to 0.38%. The predominant phenotype was central memory T cells. And if you would like to see the entire publication, please go down to the information and have a look at the entire publication. Recently, the use of CD45 array depleted memory T cells in the treatment of COVID-19 patients has been reported by Antonio Paris and his co-workers within a phase 1 clinical trial. Eligible participants were hospitalized patients with PCR-confirmed SARS-CoV-2 infection and, according to further inclusion criteria regarding pneumonia, lymphopenia, and oxygen demand. Enrollment was based on match with the HLA genotype of the convalescent donor and according to inclusion-exclusion criteria. Single infusions were applied in a dose-escalating manner. 10 patients were enrolled and 9 patients were treated. 3 patients each received 3 escalating doses ranging from 1 × 10 to the 5 cells per kilogram to 1 × 10 to the 6 cells per kilogram of CD45 array negative memory T cells. The clinical outcome of the study is described in the publication cited below. The author stated that this study provided preliminary evidence supporting the idea that treatment of COVID-19 patients with moderate to severe symptoms using convalescent CD45 array negative memory T cells was feasible and safe. This clinical application is currently being further investigated in the phase 2 clinical trial. All this and much more details can also be found in the original publication as given below. With that, I end today's webinar and thank you for your attention. If you have any questions, please ask them directly or write them to us later in an email.
Thank you again, speakers, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.